because that is they are, as soon as they are synthesized and secreted, they act and produce interaction on the nearby cells of the same cells, and then they are rapidly inactivated. Because of their rapid inactivation, they are having short half-life. The most common precursor, that is arachidonic acid. Arachidonic acid that is synthesized inside human cells, and because the source of the arachidonic acid is linoleic acid, multiple times in the lectures and tutorials this point was discussed. So major dietary precursor for arachidonic acid is synthesis linoleate. Linoleate or linoleic acid is having the same meaning. Their linoleic acid is present in plant oils or vegetable oils or the oils of vegetable origin, whatever you can say. So if you take more amount of more omega-6 fatty acid containing vegetable oil, it means you are consuming more omega-6, and this reflects the fact, or this depicts the fact that this linoleic acid is more converted into arachidonic acid, and hence this arachidonic acid is incorporated in the biological membranes, where it is incorporated as the phospholipid constituent. Now see this slide. You take dietary linoleic acid from plant oils by the action of the elongase enzyme and then desaturase. This arachidonic acid is produced and it becomes a part of the membrane phospholipid, either in the form of phosphatidylcholine or 2-phosphatidylinositol 4,5-bisphosphate. In these two forms, this arachidonic acid is incorporated. Now what happens? When there is stimulus, it binds the receptor phosphatidylcholine or phosphatidylinositol bisphosphate, either of them present in the biological membrane as phospholipid of the biological membrane or the plasma membrane. By the action of this phospholipase A2 enzyme, phospholipase A2 that acts on the phosphatidylcholine to make available this arachidonic acid. This enzyme in the presence of calcium attacks, and this is arachidonic acid is hereby available. Phosphatidylinositol bisphosphate, by the action of phospholipase C, it makes 1,2-diacylglycerol. Diacylglycerol, by the action of lipase enzyme, arachidonic acid is released, and then monoacylglycerol is also released, and by the further action of monoacylglycerol lipase, arachidonic acid is released. So these are the three arachidonic acid. By the action of their corresponding enzymes, they are available through the cytosol. And this actually acts as this arachidonic acid acts as the precursor of eicosanoids. Now what happens with that? Arachidonic acid is now available in the cytosol, and this arachidonic acid, either it goes to cyclooxygenase pathway or to lipoxygenase pathway. Through the cyclooxygenase pathway, it ultimately produces PGG2, means prostaglandin G2, that gives rise to multiple prostaglandins and thromboxans. And if it undergoes lipoxygenase pathway, then it forms hydroperoxycosa tetraenoic acid that gives rise to leukotrienes, hydroxycosa tetraenoic acid, and lipoxins. So prostaglandins, prostacyclins, that's also PGI2, that is called as PGI2, that's also synthesized in the PGG2. Prostacyclins, prostaglandins, thromboxans, they are collectively called as the prostanoids. These three and the leukotrienes are the most important eicosanoids that is included in this topic. Prostaglandins, thromboxans, prostacyclins, these three, they are called as the prostanoids. And leukotrienes, they are synthesized by the lipoxygenase pathway. Amongst these three, this leukotrienes is very important at your level. Now see, this is the slide picture taken from Lippincott, and you can prepare this topic from Lippincott. It is well explained over there. So you can prepare this topic from the source. This is adequate for your level. Same thing, phospholipase A2 action. This phospholipid is, by the action of phospholipase A2, it is converted into, or it is degraded into arachidonic acid, and the remaining phospholipid is lysophospholipid, that is deficient in one arachidonic acid or one fatty acid. Fatty acid in this case is arachidonic acid. 5-lipoxygenase, the same thing, same compound that is formed 5-hydroperoxycosatetraenoic acid, then makes leukotriene A4. This is a big flow chart given in the Lipton plot, so I made it or divided it into two portions. So this is the continuation of this previous slide. Leukotriene, first of all, A4 is synthesized, and where it is produced, leukocytes, platelets, mast cells, and heart and lung vascular tissues. So you have to learn them by heart, all of these, where they are located and what functions they perform. This is very important for the first year students. How it is synthesized or how it is metabolized, it is included in second year metabolism, lipid metabolism, eicosanoid metabolism rather. Here it is, this arachidonic acid, if it follows the cyclooxygenase pathway, it makes PGG2. How it makes PGG2? Because this arachidonic acid, that is catalyzed by, this action is catalyzed by the endoperoxide synthase enzyme, and this endoperoxide synthase enzyme having two catalytic activities. One is cyclooxygenase activity, and the second one is known as peroxidase activity. And for the peroxidase activity, this glutathione, that is reduced form of glutathione, is used as a reducing agent. And there are two cyclooxygenase enzymes. They are having two isoforms. One is constitutive and the second one is non-constitutive. Constitutive means that is all the time it's present in all the, normally in all the cells, but COX2, that is the inducible enzyme, means when there is release of cytokines, endotoxin, growth factors, or tumor promoters are there, then only then it is induced and then available in the cell. Now this, first of all, this by the action of cyclooxygenase and peroxidase, first of all PGG2 is formed, and then PGH2 is formed. Then PGH2 means prostaglandin H2, that gives rise to thromboxane A2, prostacyclin or PGI2, PGF2 alpha, prostaglandin F2. If I say PG, it means prostaglandin. Then PGE2, prostaglandin E2. So these four prostanoids, they are synthesized by the, after the PGH2 is formed, and you can see their actions. How thromboxane A2, that is produced by platelets, as I gave you example of platelets, they produce thromboxane A2. They promote the platelet aggregation, means this is autocrine action. Vasoconstriction, autocrine action means they promote the platelet aggregation. They are synthesized and secreted by the platelets, and it acts on the platelets as well in the same cells, so increasing their aggregation. Then they cause vasoconstriction, contraction of the smooth muscle. इसको कैसे याद रखना है? Because vasoconstriction and contraction of the smooth muscles are one and the same thing, and they cause the platelet aggregation. Thromboxane A2 cause the platelet aggregation होती है, smooth muscles की contraction होती है, तो thrombus बनता है. इस तरह याद रखिएगा इसको. Prostaglandin I2 or prostacyclin, that is produced by endothelium of the vessels or endothelial cells of the blood vessels. They cause vasodilation. इससे opposite होता है action. PGI2, they are, and thromboxane A2, they are having opposing effects. Vasodilation, this causes vasoconstriction, causes vasodilation. It inhibits the platelet aggregation and it promotes the platelet aggregation. PGF2 alpha, that is most important function of PGF2 alpha is that it is synthesized and secreted during the childbirth. So because it stimulates the uterine contractions. So contraction of smooth muscle, that causes the vasoconstriction. All the, you can see that it causes the contraction of the smooth muscles. Constriction of the blood vessels, that is because of the contraction of the smooth muscles of the blood vessels. So it causes vasoconstriction, contraction of the smooth muscle, and if the smooth muscle they are of uterus, then it causes the